So I'm going to give you a really whistle-stop tour of what AMR surveillance looks like in animals in the UK. We do this surveillance because we are trying to safeguard animal health, but also because AMR is a One Health issue. So surveillance in animals is really well established in this country, but it breaks down to kind of two distinct types of program. We do surveillance for AMR in sick animals, and we do surveillance for AMR in healthy animals, and they're quite different. So surveillance for AMR in sick animals, this is a clinical program. You can see here, this is a sick calf. He's sick. He's being attended by this vet. She's going to take some diagnostic samples and they're going to be submitted to a lab and will undergo bacterial culture and sensitivity. And then the results of that testing will be reported back to her and that will inform her treatment decisions. Now, so far so good, very similar to what happens for surveillance for AMR in people. But what's different and unique about the animal sector in this country is that of all these antimicrobial susceptibility tests that are performed on sick animals, a very small proportion of those tests are actually done by government laboratories. The majority are done by private veterinary laboratories. And that data, while it goes back to the vet to help her with her work, doesn't necessarily feed into government surveillance efforts. So we've got a huge knowledge gap there, and this is a knowledge gap that we're actually trying to fill to some extent through some of the work that we're doing through the National Biosurveillance Network. To move on to the healthy animals surveillance, now this is totally different. We're not looking at sick animals. We're not looking at pathogens. What we're looking at here is AMR in commensal organisms. So these are commensal organisms in the gut flora of healthy animals at slaughter. You can see we've got a really happy bunch of pigs here. These have probably ended up already as pork chops somewhere. But before they do, we are testing them for AMR. Now, there are also limitations with this program. The main one being we only do this surveillance in pigs and poultry. These are the intensively produced animal species. Historically, they were high users of antibiotics, and that's why we kind of have this historical interest in them. But it does mean that there are some other really important animal species that we're not surveying for AMR at all. Most obviously, sheep and cattle, right? These are really important sectors in the UK. They get eaten a lot. There are lots of them out there. And in terms of business as usual surveillance at UK level, we're not actually currently surveying them. And then outside of food producing animals, we also have dogs, cats, horses, other companion animals. Obviously, we're not eating them, but we do have much more kind of intimate contact with some of these animals. Where anyone who has a dog, it's licking your face every day. And this is another area where we're not doing established baseline surveillance for AMR. So another gap. And this is one that we have, again, made some progress towards filling with some of the work that's been undertaken through the PATH-SAFE program. You'll hear a lot about that in the talks that are going to follow me. But I would say it's something that we're trying to expand on because the beauty of this program, this surveillance that we do in healthy animals, is it's a representative program and it's UK-wide. And this means that we have really, really good data now over multiple years that gives us a really great indication of what AMR looks like in livestock populations, well, limited livestock populations in the UK, but also lets us monitor trends and assess the impact of interventions. So that was really high-level overview of AMR surveillance in the UK. Obviously, the programs are much more detailed and involved with lots of contributing partners. You can read the full details of these programs in the VARS report, which is produced every year. This is kind of the animal equivalent to the SPOWER report. We also have a One Health report, which is produced between VMD and UKHSA. What we're trying to do here is just shift towards a more One Health way of thinking. We provide more limited amount of data in that report, but much more contextualisation and One Health interpretation. And you can access that very easily with the QR code. Thank you very much.
Tamsin, you win the prize for timekeeping there. Brilliant. Okay, without further ado then, let's move on to Declan.
Hello. So I'm Declan Power. I'm a microbiologist. Together with colleagues at APHA, we're looking at AMR monitoring in abattoirs in England and Wales. So why sheep? As Tamsin just mentioned, Small ruminant farming represents a significant sector within UK agriculture. The 2021 census valued it at £1.5 billion. And we're a large exporter of lamb and mutton. And the consumption of lamb and meat products has been implicated in human health, hence one's health context, with various salmonella outbreaks over the years associated with eating sheep. Currently, there's no routine surveillance for sheep AMR in the UK. So this provides the limited surveillance data in the UK in the One Health context. So PATH-SAFE AIM was sort of a collaboration on top of an existing survey, which looked at different target species, sort of salmonella from Zika and carcass swabs. But that's a separate project with its own sensitivities. I'm not necessarily going to talk about that. But the PATH-SAFE aim was to take a novel One Health approach to AMR surveillance in sheep and assess the value of this data to provide contemporary data and update the risk assessment for AMR. So a bit of the overview. We had sample collection from abattoirs across England and Wales, about 12 of them, representing about 30% of the annual sort of sheep slaughter throughput, with the FSA collecting the cecal contents for us. We did microbiology, so isolating our target species of Campi, Enterococcus, E. coli, which then put on cryopreservation beads for downstream analysis of MICs, whole genome sequencing, and then the analysis and reporting, which is ongoing now. So the sample collection, the sampling kits were already assembled and sent out from APHA Weybridge to each individual abattoir site. They included anything that they sort of needed to do the sampling, so pre-labeled pots with all their reference IDs, sort of biochills, gloves, scalpels, and we told the FSA to collect at least 30 grams of cecal contents from healthy lambs and ewes at the point of slaughter. So the sampling numbers were representative of the abattoir throughput. So larger establishments had more samples and smaller, fewer. And the samples were then shipped to us within 48 hours of collection. Typically the next day we got them and we processed them for the microbiology. And we received samples from February through to October in 2023. So the microbiology lab methods, we had direct and enriched cultures for our target species. Very briefly, for Campi, we put a loop of Zika onto selective agars, so MCCDA and Butzler, and then confirmed presumptive colonies with Molditoff. And then for AMR, it was enriched in buffered peptone water, and Monconchi with and without cefotaxime, and carbaroxyplates for carbapenemase, and confirmed with indole oxidase testing. And for Enterococcus, BHI, Slenter barley, and again, confirmation with Molditoff. So the MIC, we use a sensitizer system, which is sort of semi-automated, high throughput, with commercially produced plates with a panel of antibiotics, which we read as the lowest concentration that inhibits growth. And we determine our susceptibilities with the ECOFs. So we use 4 different plates. We use UCAMP for Campi, UVENC for Enterococcus, and UVSEC 2 for the AMR presumptive colonies, and UVSEC 3 for the background E. coli. For the whole genome sequencing, we use a subset of isolates to send to APHA Weybridge sequencing unit, extracted via Kingfisher and sequenced on a NextSeq Illumina platform. I've shamelessly stolen this figure from Manal, so she's going to go into more detail later. This is the whole genome sequencing side. Another proportion of this sort of work stream was a wastewater and environmental sampling of the abattoirs itself. So the current methods of cecal and swab sampling are quite invasive and take time away from the Food Standards Agency, the hygiene inspectors, people who are doing the sampling itself. So we're looking to use more pathed methods of surveillance, which might be easy to implement in the long run and free up a bit of time for the everyday workers. But as yet, their performance is sort of unproven. So we're running pilots to determine the viability of these sampling methods. Namely, we're using environmental swabs, so swabbing any sort of floors, surfaces, railings, whatever we can find within an abattoir, and putting those through the same sort of methods I've just discussed for microbiology. We're using more swabs, which is one of these. It's like a little bit of gauze tied with a bit of fishing line and a hook on. We can suspend that in a floor drainage system of an abattoir, and any sort of runoff, any drips that sort of get washed through, theoretically collects the same that we can get from an environmental swab. And more recently, we've been collecting wastewater in collaboration with CFAS, So post-screening, we've taken wastewater samples and they're currently being processed by CFAT. So this pilot was to explore the sampling methods to augment or replace the existing approaches, as mentioned. As yet, we've done sort of 2 abattoir visits of 2 different species, so 2 to pig and 2 to sheep. We've stored down our isolates and we're waiting to do further analysis. Same again, so we've got MIC testing, whole genome sequencing. So the hopeful outcomes of this is to provide a surveillance baseline for potential zoonotic pathogens, not just Campi as it says here, and a previous unexplored One Health compartment, and provide that baseline for the AMR of the organisms that we have recovered. And this can be used to inform and evaluate the risks of zoonoses and AMR transmission from consuming meat to us as the consumer. And this supports the UK-wide National Action Plan for the control of AMR. And the data collected in the isolates we've obtained can be archived to support future work that we have going on. So the future directions with the continuation approved, we're looking to continue this AMR surveillance with these alternative methods. So build upon the pilot data that we've got so far with more abattoir visits, like re-establishing that these methods can be used to reliably represent what we're isolating from seeker and carcass swabs. And so we want to refine the protocol and create a sort of standardized method that we can roll out to FSA staff and assess the feasibility of the FSA staff to undertake this sampling for us, and eventually conducting a larger pilot study to reinforce these sort of methods. And with that, that's short and sweet. Any acknowledgements? Sorry if I've missed anyone off, but particularly bacteriology, Natasha, Graham, and Al helping out with the sequencing of these sort of isolates. Francesca for project oversight, VMD, Andrew, Tamsin, and all the industry people that's helped participation from the abattoirs and also allowed us to come to their abattoirs and visit. I know it's been not that well received in certain places. Yeah, thank you.
Thank you, Declan. We're going to move straight on. I should have said it, just as with the previous session, we're going to hold all questions to the end and do, or everyone in one group at the end. So I'm going to pass it straight over to Sue.
Thank you. I want to tell you a short story, lacking on scientific detail, but hopefully highlighting what can be achieved with that sort of collaboration and cooperation that was being mentioned earlier. So the story starts up in a SRUC outpost in the north of Scotland with a small team of vets, epidemiologists, lab people, and my co-lead, Jeff Foster, who's a microbiologist, bacteriologist, and who runs the healthy monitoring surveys that are run in Scotland for AMR on 4 species that have been done since 2017 and are in the SONAR report. So once upon a time, but it was only 18 months ago, we were asked by VMD on behalf of the PATH-SAFE programme, could we potentially run a UK-wide AMR survey of cattle to fill that gap, healthy cattle at slaughter, in accordance with EU harmonised requirements, and complete the report by March 2024. So when I picked myself up off the floor laughing, I said, no, that's not possible. What we could potentially do after a bit of discussion was we could run a 6-month survey, and we could try to do that, but it would be hugely dependent on not only the goodwill of the agencies involved, but engagement from the meat processing industry, because they would have to give us access to their premises, because this is a voluntary initiative. Potentially logistical nightmare, still hugely ambitious, but we thought we would give it a go. So in the beginning, some of the hurdles and challenges were that there were a lot of processes to develop in a very short time. For instance, how do you get cow cecal content samples into your very small pots within a food producing environment that's heavily regulated. We were working with 3 different regions of the UK, all with slightly different agencies, with different working structures, different relationships with premises, different lines of communications. And I have to thank the VMD and PATH-SAFE people for putting us in contact with the right people and facilitating that. We had to work out how many samples we were actually going to take to get the target number of isolates of the bacterial species required, when there was no real previous information on that. So we were very reliant on Jeff's long-term and years of experience and knowledge there. We had to work out a sampling strategy proportional to throughput by region and by premises. And that's not as easy as it sounds when the data is not necessarily available to do so, and the structure and the granularity is different across each of the regions. And it's totally dependent on which premises you can get to participate. And that was one of the real big areas that in future we'll need to work on a lot more for these sort of things. As you saw, they got 30% representation for the sheep. We probably achieved the same for the cattle. But you're working with an industry that, in the second presentation, it was mentioned about misinterpretation of data and miscommunication, and they have reputations that they're concerned about. We are extremely reliant on the goodwill of these people to do this, because it's voluntary. And that needs time to build awareness of what we're trying to do to get them engaged. And we didn't have that time. So a lot of making friends, influencing, a bit of workarounds, lots of logistical constraints, and too little time. And I'm absolutely impressed with the enthusiasm of everybody we've worked with and the can-do attitudes here. So we had a plan. Plans go astray, but we didn't go too far astray. There were a few minor slippages, a couple of teething problems, occasional rescheduling, 2 major issues. Despite an awful lot of effort, we couldn't get the Northern Irish industry enabled on board to us to be able to sample in their premises, so it became a British survey. We had lab capacity issues. Our new building wasn't ready, and so we had to find workarounds for that. At the end of December 2023, we had the samples that we had requested from Scotland and England and Wales. We had the bacterial isolates identified. We still had the MIC testing to do, and the analysis and the reporting, all by March. And it's a short quarter in the year. We had light at the end of the tunnel, and we might just make it. So that's where we were. And then, thank you guys, they asked us for some more. Could we do the extra that we hadn't got from Northern Ireland? And could we do that in Great Britain? The premises and the FBOs we were working with were very, very positive. The field teams from FSA and FSS were very positive. And we in SRUC were going, because we were moving into a new building, we had no labs, etc. So we had to explore several options to move the actual processing of the samples. And ultimately, we're doing that with APHA at the moment. And that's meant minor changes. So the field teams are taking those in their stride, which is absolutely great. We will be reporting on the basic results from the original survey by March. Obviously, the rest is going to take a little bit longer. There have been a large number of lessons learned. This does serve as a really good pilot. We now have some idea of the types of frequencies of some of these bacterial and the species and the subspecies. And we also, you'll be saying, this is PATH-SAFE, what about sequencing? we are sending the isolates off for sequencing. And last week we heard that we had been awarded a very prestigious local East Bio place for a candidate on their postdoctoral program starting in October, who will be able to work with that sequencing and that analysis. And that too is down to VMD and FSA for helping us put together a case studentship there. So a big thank you to absolutely everybody involved in this, because we couldn't have done it without any one of them. But a really, really big thank you to the industry, because none of this would have been possible without them letting us go into the premises to take the samples. And it's been a real pleasure working with everyone. Thank you.
Thank you very much, Sue. And finally in this session, we have Manal who's doing a double act, I think, two for one talks in one go, and then we'll take some questions. Over to you.
Okay, hi everyone. I'm Manal Aboon. I'm from the Animal and Plant Health Agency, and we've been working on two projects. I'm first going to talk about looking at AMR in bulk milk. So you ask why look at AMR in milk? So milk is usually collected from the cows and then combined on farm and then stored for a period of time until it gets collected by the milk processors. And so during this time, when the milk is collected, it could be contaminated with fecal contents from the cows or environmental contamination, and that's how resistant bacteria get into the milk. And then so in these tanks, they're waiting there for maybe a couple of days. You need to check the quality that the milk is good and meets the safety requirements, standard requirements. And so one of the things that the national milk labs do is look for various indicators of quality of the milk. So looking at general bacterial counts, and also looking at the presence of antimicrobials within the milk samples. They also then do some testing to look for mastitis pathogens in the milk as well. And then currently there's very limited AMR testing, and it's usually for by PCR of a beta-lactamase gene. So looking at milk, which it's usually processed and pasteurized, so they reduce the amount of bacteria in there. But there are some parts of the UK that do drink raw milk, and so that could be a possible route of transmission for consumers. And apparently some people bathe in milk, but I don't know why you would do that. So working with National Milk Records, which also have laboratories, they are an organisation that work with the producers, with the vets, with different government agencies, and they look after the milk sector. And so they produce reports, and they look at status of the health status of animals in the UK. They also provide a scheme called Farm Assist, where they where they look at the antimicrobial usage in cows, and then they've monitored that over the last few years, and they can see when antibiotics are being used and reduced. And so they have two laboratories, one at Wolverhampton and Glasgow. So we worked with the guys at Wolverhampton, where they do the range of tests that you see listed there to look at the quality of the milk and to ensure that it's safe for consumption. So as I said, so the milk is collected by the farmers, and then collected from the cows by the farmers, and then the milk processor will then collect the milk and then send it to the labs for statutory testing. So just move to the next one. So sorry. Yeah, so okay. So the aims of this project then were in the 12 months that we had is to look at the occurrence of AMR bacteria in milk, because that's not really been looked at as thoroughly before. And we collected milk from different areas in the UK, and that was done by proportionally. So where there's lots of milk being produced, we took more samples throughout the year. And then as part of this study, as well as isolating bacteria, we also did some whole genome sequencing of that, but also qPCR to try to develop a qPCR that would be able to detect this class 1 integron. Class 1 integrons, for those that don't know, are normally associated with AMR genes. So it's an indicator that you could possibly have resistant bacteria in your milk sample. And now move on. Okay, so this is kind of roughly how everything worked smoothly, as Sue was saying earlier on. So at the very beginning, it was APHA, VMD, as well as the National Milk Record, putting agreements in place and kind of coordinating sample collection. Then the National Milk Labs, they would send us, the samples weekly for testing, and they would go down to Starcross. So APHA, for those that don't know, we've got multiple labs around the country. So the milk was sent to Starcross on this occasion. And down there, they would do the bacterial isolation, and then they also did the phenotyping, so MICs. And then the samples were then sent to Weybridge for mouldy, for identification of particular species, and the sequencing, as well as the integron PCR. Okay, so to give you a bit more detail. So the National Milk Labs, they're the ones that got permission from Arla Foods. So without them, we wouldn't have milk. So they supplied us with the milk samples, and it was NML that coordinated the collection monthly from the different regions. And they would send them 5 mils of milk to Starcross for the bacterial isolation, and then monthly they would send us a batch of 100 samples to do the milk testing, and they'd come looking like that. So samples would arrive usually on a Friday, and the testing would start on a Monday after they've been in the fridge. And then we've done that between January and October. Been quite intense. So then the microbiology, a little bit like Declan showed a little bit earlier, they would do direct plating, looking for Streptococcus, Klebsiella, E. coli, Staph aureus from the milk samples, and then to look for specific resistance, we would use enrichment. So that's normally what we do when we look at surveillance of pigs and chickens. And yeah, so then after enrichment, we'd have the colonies that we were looking for on whichever plate the target would grow on, and then they would be picked and stored and sent then for MIC and sequencing. So the MIC, for those that don't know, so we did that at Starcross. It's the not Kingfisher, sorry, Sensitita system from Thermo Fisher, and this one comes with a standardized plate that's already includes your antimicrobials at different concentrations, and we use that to test the different bacteria to collect their MIC values. And then the MICs work whether it was resistant or sensitive would be determined using epidemiological cutoffs. So that's looking at baseline. So we weren't using clinical cutoffs that you'd use in a human lab. Then in terms of the sequencing and also the characterization, so with Enterococcus in particular, it's quite hard to see the difference between the two species that we were after. So that's where we used MALDI-TOF to look at basically the protein signature of the different species, and we just looked at Fecium faecalis. And then all the samples were then, or most of the samples, were whole genome sequenced, and then they followed this basic pipeline. So using Kraken to look at the bacterial identification to make sure E. coli was E. coli and not something random. And then we do assemblies to join basically the short read data together to look for plasmids and where AMR genes were. We also did some sequence typing to see if there's any common genetic backgrounds in the milk samples. And then we used in-house tool called APHA Sect Finder along with Abricate to identify AMR genes in the samples. AMR genes and plasmids where we can. It's not always possible with short read sequencing. So hopefully in the continuation, now it's been agreed, we'll do some long read sequencing to see what plasmids and genomes we've got out there as well. And then at the end, we try and the plan is to do some genotypic and phenotypic correlation. So hopefully if you have a gene, you should be resistant. If you don't have a gene, you should be sensitive. And for some bugs, that's easier than others. So that's going to be something we learn as well. Okay, and then to put this kind of all into some sort of context, the epidemiology guys at APHA are going to be looking at using the AMR data to look at the prevalence of resistant bacteria in the milk population, and are there areas in the country where you've got higher or lower AMR genes, which we could then possibly link to AMU usage as well. And I think that's it for milk. So I just want to say big thank you, obviously, to ALA and NML for the samples, because without ALA, we wouldn't have milk, as I said. And without NML and NMR, sorry, I think Eamon's here somewhere. I can't see you in the crowd, but they did a lot of organisation at their end so that we could do the lab work. And then the guys at Starcross, they've been super busy down there, receiving samples and processing them, and obviously the guys at APH at Wadebridge, sorry. And then there's a whole lots of people that helped oversee the project, and obviously VMD and PATH-SAFE for supporting us and funding. So I'm going to carry on. The slides look kind of similar. I was hoping they wouldn't be so close together, but hey ho. So I might skip over a couple of them so we save a bit more time. So animal feed. So animal feed, we grow it in the country, but we also import a lot of it as well. And so that is a possible route of transmission. And currently, sorry, that's a route of transmission. And with over the last 10 years that we've been looking at usage in farms and the AMR in farms, antibiotic use has gone down, AMR levels are reducing as well. So there could come a point where the stuff that we're bringing in is actually making the AMR, well, not unable to go down or maybe increase it. So there's currently no testing of antimicrobial resistant bugs in animal feed, whether it's raw or a finished product, and whether it's imported or not imported, apart from the salmonella that's being tested. And for those that don't know about animal feed, I didn't know about animal feed until this point. So you get the raw ingredients, so wherever that comes from, whether it's the UK or outside, it gets milled. There's different processes to do that. It gets mixed. If it's a finished product, there might be 10, 20 different products that are going in there being mixed. And then there'll be different lines of feed that are given to chickens or cows or whatever the animal that's being fed, and some of them get heat treated, some there's no heat treatment, so there's all kinds of different ways of looking at it, of way it's produced. Sorry. So there's a couple of recent studies in Sweden and US that looked at AMR in animal feed. So this hopefully will add to that knowledge base. So we didn't do this without a lot of help from the Agricultural Industry Confederation, which is basically a trade organisation that cover quite a big proportion of the animal feed sector and various other things. I don't work for AIC, I work for APHA, so I'm giving you information that is I've been given. So they support commercial agriculture in the UK, and they help livestock farmers with giving news and information about new products and feed additives, and they try and ensure that the feed that they're producing is sustainable as well. And then they implement some regulations and animal health and welfare considerations as well. Okay, so the aim of this particular PATH-SAFE project of AMR and animal feed was to work with a feed mill in the UK that are producing pig and poultry food. And the idea was to basically look at what bacteria are in these samples and are any of them antibiotic resistant. So we got a selection, so about half of the samples were from raw feed ingredients and the other half were finished products. We collected the isolates, we've done some genomic characterization, and well, the aim is to find the prevalence and what's the risk of these feeds introducing AMR to the livestock. So it's just a similar overview, except that the feed mills would send us 10 samples a week of each of the different types. And these samples actually went to the APHA in Newcastle, which actually this was a relatively newish type of work. They used to do it before, but they hadn't done it for a while. So they actually quite enjoyed doing this project, which is a good thing, right? And then the MIC testing went to Starcross, and the sequencing was at Weybridge. And it's much the same. So the feed mill, at the feed mill, the APHA staff went at the very beginning to help train the staff there on how to collect the samples, and obviously being really careful about cross-contaminating anything. And then weekly, we'd get 25 grams of each samples, which you can see there in the bottom right, I think. And at the top, you can see what the different types of feed that we were sent. So there was the raw ingredients. I think most of it was barley and wheat. And then on the finished products, there was several different lines included. So in total, we had 300 samples of raw ingredients and 300 of finished products. And then the isolates that were recovered were then sent for either MIC or sequencing. So this was a little bit different, I guess. So for all of these, they ended up in enrichment. I'm not sure if we were expecting to see lots and lots of different bugs in there, so hence why in the enrichment. And then we used a range of different selection plates to identify the various different bugs that we were after. So colonies were selecting. There's some examples of what we're trying to picking. So as I said before, Enterococcus, faecium and faecalis on a plate, they're just pink on a plate, which is not very helpful. So that's why we were doing moldy to identify which the target species. And I will. Okay, so this is much the same as what I've just talked about a few minutes ago, so I won't go over this again. Unless someone asks me later. And then there was MIC testing, which I think I've already done. So I will skip over this one as well. We used basically the same plates. There wasn't much difference. And then, so in terms of the epidemiological analysis, so we're going to see if there's a higher, or what the prevalence of AMR bacteria are in the different feed products, whether it's the raw ingredients or the finished products. And we'll look at we're going to try and compare what we're finding in pig and poultry isolates direct, and compare those to what we're getting in the animal feed. Are they the same? So if they're not the same, that's not where the animals are getting their resistant bacteria from. And big thank you to AIC and Simon Williams for basically assisting us in getting the samples, because it's not something that we've done before, looking at animal feed. And then the guys up at APHA Newcastle, Starcross again for the MICs, and then the sequencing and all the isolation and the epidemiology stuff that's been happening at Weybridge, and the oversight as well. And that's where I stop.